Well, hello, Google. Thank you so much for having me. It's brilliant to see you all here. I'm only slightly disappointed that there's no dogs in the audience, because that would have been completely awesome. But maybe next time I have a book, you can all bring your dogs in. That would be amazing. So, yeah, I'm here to talk about my book, Bring Back the King, The New Science of De-extinction, which is all about, as Kit said, the ability of scientists to bring extinct animals back to life. And it's a science that's very much in development. It's not quite with us now, but I think it will be with us reasonably soon. And this, as you can see, is the book. Here is my dog, Higgs. He's modelling the book there for you, just to give you an idea of sample copies. He's called Higgs the dog particle, which is a joke that you guys at Google get, which I completely love, because people would normally look at me and stare blankly at that point. So that is Higgs before he read the book, and oh, man, that is Higgs afterwards. I think he found it quite satisfying, quite exhausting. So he's my promo dog, which is good. So the book asks, which animal would you like to bring back? And I'm doing an event at New Scientist this weekend, and we put out a thing on Twitter just to gauge people's reaction to this. We said, what animal from the past would you like to resurrect? Just to get a feeling of where people were with this topic. And sample answers included the great auk, good example. Smilodon, which is a saber-toothed cat, interesting. And somebody said, I'd like a pet velociraptor, please, which kind of intrigued me. So I tweeted them back and I said, why a pet velociraptor? And the guy said, because I fancy a pet with retractable claws. Which is kind of interesting. So I tweeted him back and I said, how about a cat? And he tweeted me back and said, I'm not a cat person. So there we are. And you'll notice also I've anonymised these tweets, clearly. I'm not just followed by people that have guinea pig avatars. So there we are. Other sample answers, just quickly before I moved on, include, can they make a half-man, half-woolly mammoth? Which is interesting, because I think I've been living with that particular thing for about the last 10 years. He's called a man-off. Do you see what I've done there? Somebody else said, Margaret Thatcher clones, thousands of them, millions, an army. Slightly disconcerting. That's not what de-extinction is about. And then somebody hit the nail on the head almost when they said, dodo. Then I'd farm them and start a fast food chain. Dodos, KFD, Dodo Cottage. Which was a good joke, but was topped when somebody else tweeted, Nandodos. Genius, absolute genius. Okay, so let me tell you about de-extinction. I want to start by telling you a story. And it's a story about a goat. But this wasn't any usual kind of goat. This was a goat that lived in the Pyrenees thousands of years ago. And these were amazing animals. They were big, stocky beasts. They weighed about as much as a washing machine, but they were much more agile. And they had these enormous, great big, curved horns. And what they used to do is they used to spring around on the mountainsides, casting impressive horned silhouettes against the clear, blue, cloudless sky. And they did that brilliantly for thousands of years, until we came along at the end of the 19th century. And we looked at them, and we looked at their horns, and we looked at our empty living room walls. And you have to remember, this was in a time before IKEA. So there was nowhere to go to buy reasonably priced pictures or frames. So our walls were empty at that point in time. So they looked at the Picado's horns, they looked at their empty living room walls, and they thought, do you know what? That would look really good there, which is a shame. So we started hunting them, and their numbers plummeted. So then if you fast forward to the year 1999, there was only one left, one Picado. And that was an elderly adult female called Celia. And scientists knew that her time was running out. They knew they had to do something. So they went up on the mountainside and they caught Celia and they put a radio tracking collar around her neck. And then they let her go. And they kept an eye on her to see what she was up to. And then the next year, the radio tracking collar let out an ominous beep, beep, beep. And it was the signal they had been dreading. They rushed up the mountain to find her, only to discover that Celia had been crushed by a falling tree. Can I have an R for Celia? Thank you. So the Bucardo was no more. It had ceased to be. It was an extinct Bucardo. And that was it. And that should be it. That's what happens. When animals go extinct, that should be the end of it. Except that increasingly, that doesn't have to be the case anymore. So actually, if you rewind a bit to the point when they put the tracking collar around her neck, they did something else, the scientists. They actually took a couple of biopsy samples from the animal, and they carted them away, and they froze down her cells in liquid nitrogen. And then a couple of years after she died, they thawed these cells, and they did the kind of experiment with them that a few years earlier had been used to make Dolly the sheep. Now, are you guys familiar with Dolly the sheep? I'm guessing you are. So here she is. I just want to sidetrack and talk about Dolly for a second. So Dolly the sheep was, of course, the first mammal to be cloned from an adult cell. And she showed that you can take the DNA from an adult cell, add it into an egg that's had its own DNA removed, and then use that to create life. And that was phenomenal. So Dolly became this huge media sensation. The paparazzi literally followed her every move. So here she is on the front cover of Nature magazine shortly after she was born. Then here she is a few years later on the front cover of Hello magazine. And then a few years after that, sadly, on the cover of Waitrose Food Illustrated with a little rosemary and garlic.
The real is the meal made from the sheep?
No. No, in fact, where she's ended up, she's stuffed and she is in a museum in Scotland. That's her final resting place. God rest her little fleecy soul. So that was the experiment that inspired the Bucardo scientists, okay? So they did the same kind of experiment. And then in 2003, after hundreds and hundreds of attempts, something incredible happened. And this is the actual moment when it happened. So they created a little baby Bucardo, Celia's identical clone. So this is the moment that de-extinction actually first happened. And this is actually over 10 years ago, which most people don't realise. So it was a phenomenal achievement, phenomenal. And she was beautiful, this little clone. She was gorgeous. She had tousled brown fur and wonky legs and toffee-coloured eyes. But unfortunately, she developed breathing problems, and then she died just a couple of minutes after she was born. So the Bucardo isn't just the first animal to become de-extinct. It's also the first animal ever to go extinct twice, which is a bit of a shame. But I spoke to the scientists who were involved in this particular piece of work, and they absolutely assure me that with time and with funding, those are the two limiting factors, they are completely convinced they will be able to bring the Bucardo back. Not just one, but many healthy Bucardos that they'd like to see released into their former range. So I don't know how that makes you feel, but that story leaves me with mixed emotions. I feel pretty sad that we treated the Bucardo so badly, but I feel really excited for what might be possible. And I want to show you my favourite photograph of me ever. So this is me on the beach at Mammoth, as Kit mentioned. And I'm the little dude on the right-hand side in the awesome red welly boots. And I'm about 2 years old. And we used to go down there fossil hunting. And the rocks there are 198 million years old. They were formed at a time when there were huge, great big reptilian sea monsters in the sea, pterosaurs in the sky, and dinosaurs on the land. And it made me dream about bringing back a dinosaur. So when you think about de-extinction, you think about what might be possible. And I begin to wonder, could you bring back a dinosaur? And if you did bring back a dinosaur, to my mind, there's only one contender, and it's this, T-Rex. It has to be a T-Rex, right? Once described as the absolute warlord of the jungle. Its teeth have, in the scientific literature, quite humorously been described as lethal bananas. So if ever there's two words that don't go together, I think it's lethal and banana, or possibly Trump and White House. Also quite bad. So I was really interested, can you bring a dinosaur back? So I spoke to the scientists, and they tell me it boils down to one raw magic ingredient, which is this: DNA. And here is some that I knitted earlier. So DNA is a very beautiful molecule, but it's very flimsy, it's very fragile. And after we die, it begins to break down. And in exceptional circumstances, it can be preserved for a long time. But the oldest thing we have ever extracted DNA from, meaningful amounts of DNA from, is less than 1 million years old. So dinosaurs died out 65 million years ago. So no DNA, no dinosaurs, sorry. So we're not going to see a dinosaur. The closest thing we will ever get to a living, breathing dinosaur is this, which is my dog in a dinosaur onesie, shortly before he called the RSPCA. So that's dinosaurs. The other question I get from people is, how about these? How about dodos? And I have to say, these beautiful illustrations which are in the book are the work of a guy called Matt Dawson, who is fantastic. And so this is his dodo. Could you bring a dodo back? Now, they died much more recently, so in theory, it might be a bit more possible. However, where they lived in Mauritius was very hot, and the soil where they fell when they died was very acidic. So their DNA is not preserved. So no DNA, no dodos. Sorry about that. But it got this idea going in my head when I was thinking about the book, and I thought, so you can't do dinosaurs, you can't do dodos. You need something from much more recent history. How far could you take this technology? Where could you go with it? And I began wondering about this man, Elvis Presley, a man deemed so sexy by the TV stations of the 50s that they refused to show him from the waist down. He was too damn sexy. And I began to wonder, could you? I don't think it's a good idea. I am not saying that we should do this, but as a thought experiment with the technology we have, could this be possible? And then I came across this website. Genuine website, Americans for Cloning Elvis, founded by a guy called Bob Mayer, who I spoke to for the book, alarmingly banned on Facebook. And they have a petition you can sign if you think cloning Elvis is a good idea. And quite a lot of people have signed it. How serious they are, I don't know. Anyway, the good thing about this website is it gives you a couple of ideas of starting places for Elvis's DNA. Because if you want to bring back Elvis, you need a source of his DNA. So the first possible source of DNA they recommend is this. These are toenails. So there's a lady called Joni Mabe, and she runs a museum in America. And she was going through Gracelands shortly after Elvis Presley died, and she went through his man cave room, his man den, and had a carpet like this, but dark green, shag pile carpet. And she kind of got down to her knees at one point and started just feeling around the carpet, maybe a bit odd, but hey. And her finger snagged on something, and she picked it up, thinking it was like a wayward sequin or something. And it was dark, so she popped it in her pocket. And when she got outside, she discovered it was a toenail. Now, toenails are good sources of DNA. They preserve it really, really well. But you guys are probably ahead of me. This isn't an obvious choice for bringing back Elvis, because what if it didn't belong to Elvis? What if it belonged to some American tourist wearing open-toed sandals? It's an awful lot of trouble to go to. So we scratched that off the list, but they have another source of DNA. And this is Joni Mabe's prized possession in her Elvis museum. It's the Elvis wart. So in that little test tube there, it's a test tube of formaldehyde, and it has a wart. Now, Elvis Presley used to have a wart on the back of his hand, and before he went into military service, the wart was removed, and his doctor had the foresight to save it. This is in the days before eBay, so it was quite forward-thinking. And then it was purchased by the lady from the museum, and I thought, well, maybe we could get DNA from this. And we could get DNA from this. But warts are caused by viruses. So this DNA is going to be a mix of human and viral DNA. So we don't want to go there either. So I kind of ground to a halt, and then I thought, well, hang on a minute. How about this? How about Elvis Presley's famous, iconic quiff? right? Hair is a really, really good source of DNA. What would happen if we did that? And I went onto eBay out of curiosity, and I put in the phrase, celebrity hair, because I was intrigued to see what would come up. And it's really interesting if you do that. If you put in the phrase celebrity hair, you can buy locks of hair that belong to all sorts of people, including Abraham Lincoln, Princess Diana, and Michael Jackson, presumably from before the Pepsi commercial. Don't know if you remember that. You can buy hair from Elvis Presley, and I'd just like to read you, if you'll humour me, a small excerpt from my book of what happened when I actually bought a sample of Elvis Presley's hair on eBay. So, this is chapter 6, The King of Rock and Roll. It goes like this. When it finally arrived, I was more than a little disappointed. Sometime earlier, I had bought some of Elvis Presley's hair on eBay, and like a child waiting for Christmas, had then had to endure weeks of overexcitement and speculation while I waited for my purchase to turn up. I was expecting something as iconic as the king himself. The package, I told myself, would at the very least be made of gold lame and studded in rhinestones. It would arrive with great ceremony, serenaded on by its own backing band, willed on by the frenzied scream of female fans who would camp outside my house and faint when the parcel arrived. But when the postman finally delivered my precious cargo, it was without fanfare. He didn't even ring the bell. He just popped the plain brown envelope through the letterbox and walked away. I picked up the drab, un-Elvisy anticlimax and turned it over in my hands. Save for the Memphis postmark smudged across the top right-hand corner, there was little hint as to the import's legendary contents. Perhaps things would be more promising on the inside, I told myself. Brimming with nervous energy, all shook up even. Do you see what I've done? I carefully opened the envelope and teased out the contents, but the luxuriant quiff of my dreams was nowhere to be seen. Instead, I found myself staring at an A4-sized certificate of authenticity. You are now the proud owner of a genuine strand of Elvis Presley's hair, the certificate crowed. But where was the hair? Perhaps they had forgotten to include it. Or maybe it was being sent separately under armed guard from Graceland. I scoured the shoddily photocopied piece of paper for a clue, and it was only then that I noticed a blob of dried glue at the bottom of the page. Inside it, like an insect trapped in amber, was a short strand of hair. I stared at it, unable to believe what my eyes were seeing. Incapable of processing what I held in my hands. Words actually failed me. Not only was the strand minuscule, smaller even than a single eyelash from my face, it was also, and I can't bring myself to say this, ginger. Elvis Presley, if the certificate was to be believed, was strawberry blonde. As the penny dropped, I felt so lonesome I could cry. I had spent $14.90 and endured a longer wait than the King's fans had for his comeback tour, only to receive what appeared to be Ronald McDonald's nasal clippings. So I did the only thing I could think of. I sealed the certificate back up in its original envelope, found a thick black marker pen, and wrote upon it, "Return to sender," adding that the address was unknown. The irony, I was sure, would be lost on them, but it put a smile back on my face. Then, bright and early the next morning, I sent my letter back. Oh, one person. Thank you very much. So listen, Elvis Presley. I was so intrigued with this idea, I phoned up a scientist who works in de-extinction, and I said to him, "Can you de-extinct Elvis Presley? Tell me." And he thought about it for a while, and then he said, "Yes. People have been cloning pop stars for decades." And I said, "You know what, you're right, obviously, because Simon Cowell, he produced One Direction, didn't he?" And the scientist said, "Yes, and that is the reason that human cloning must never be allowed." So it must never be allowed for all sorts of genuine, ethical, moral, health and safety reasons. And also because it leads to the production of songs like You don't know you're beautiful. So with that, I put the Elvis idea to bed, okay? De-extinction is not about bringing back dinosaurs. It is not about bringing back the king of rock and roll. That was just my whim, my folly. And then I started exploring de-extinction more deeply. And that's what I want to talk to you about now. So there are genuinely about half a dozen de-extinction projects that are ongoing around the globe now, trying to bring back different endangered species of animals. This is one of them. This is an animal called the gastric brooding frog. And what you've got here is a big frog with a little frog in its mouth. Now, the big frog isn't eating the little frog. It's actually giving birth to it. So this is a frog that was discovered in 1973 in the babbling brooks of Queensland, Australia. And what would happen is the female would lay her eggs, and then the male would come along and fertilise them, but then the female would eat them. She'd swallow them, they'd pass into her stomach, which she would convert into a makeshift womb, and then the eggs would hatch, turn into tadpoles, turn into froglets. She could have about 20 in her belly at any one time, and when the time was right, she would literally butt them up. Literally. And this is the subject of a de-extinction project in Australia under the guidance of a guy called Mike Archer. And the guy who kept these animals in his lab in the 70s before they went extinct had the foresight to save some body parts in the freezer, and they're using those for cloning. So that's one project. This is another project that you're all probably a bit more familiar with. The rather wonderful woolly mammoth, that iconic species from the last ice age. Now, the woolly mammoth is a good candidate for de-extinction, maybe because we have plentiful sources of its DNA. It's quite commonplace to find tusks and teeth and bones in the melting permafrost of Siberia. And sometimes, scientists come across stuff like this. Okay, so this might not look like much, but this is actually pretty much the entire torso of a woolly mammoth that was found in a remote Siberian island in 2012. And when they looked at it at autopsy, they realised that not only did it have fur and hair, it had skin and flesh and muscle. And when they cut into it, I spoke to one of the scientists who was at the autopsy, it said it looked like it had fresh meat. It looked like it was a fresh beef steak. So well preserved was it. This animal was frozen continuously for the last 40,000 years. And there are 3 different groups of scientists trying to bring back the woolly mammoth. And the first 2 are hoping to use carcasses like this, get cells from them, and use the DNA inside those cells for cloning. Right, and that's an amazing thing to try. However, as an ex-cell biologist myself, I'm slightly skeptical, because these mammoths have been frozen for a very long time with no kind of protective chemicals to protect the cells like we would do if we were freezing cells in a lab. So the cells shatter, the DNA leaks away. It's quite a tall order. But fortunately, there is a third group who are trying to bring back the woolly mammoth. And if I have to put my faith in one particular group, it's this group. And it's led by a guy called George Church, who works at Harvard. And my faith in George Church is based on 2 particular reasons. The first is that he is a remarkably intelligent guy. He has a brain the size of a planet. He is so innovative. He has this amazing genetics lab, kitted out with all the latest stuff, and an army of people working for him who are equally as brilliant. And the second reason is that he has this enormous grey bushy beard. So he looks a lot like God. So if I'm going to put my faith in one group, it's going to be the one led by the guy that looks like he would belong on the roof of the Sistine Chapel. And he's not trying to clone a mammoth, he's trying to do something different. So what he's doing is he's getting elephant cells, and then he's editing mammoth genes into elephant cells. And he's using this technique of the moment that everyone's buzzing about called CRISPR-Cas9, which enables scientists to edit genomes with pinpoint precision. So he's taking the genes that made the mammoths mammoth-like and different to elephants, genes that controlled the growth of their fur, their big, thick rolls of subcutaneous fat, and their special hemoglobin that ferried oxygen around at low temperatures, and they're putting them into elephant cells, and they're going to use those to create life. And I wish him well. And a third project is this. It centres around this bird, the passenger pigeon. Now, this is a bird that's really well known in America, where it used to live before it went extinct. It went extinct at the beginning of the 20th century, but before that, for tens of thousands of years, the passenger pigeon existed in vast, mind-boggling numbers. It was a beautiful, athletic bird, and a flock could contain billions of birds. A flock could be 100 miles long and a mile wide. It could take days to pass overhead, and when it did, it blotted out the sun. They were like an avian eclipse. And then one day they were gone. Pardon me. And the scientist who's trying to bring them back is using this same gene editing technology to try and do that, and some very clever cell biology. So there's 3 of the projects that are ongoing, but of course the most important question is this. Why? Why are scientists trying to do this? What is the point? Right, and that is it's the most important question to ask. And there are lots of reasons why, personally, I think de-extinction might be worth a shot. The first is this. If you think about the animals we've just mentioned, think about the causes of their extinction. The gastric brooding frog went extinct because it suffered from something called the chytrid fungus. And in fact, amphibians all around the globe are being affected by this fungus, and there's a massive amphibian die-off going on. And we're spreading that fungus around the globe through the commercial transport and trade in amphibians. Humans were implicated in the demise of the woolly mammoth. It may not have been exclusively our fault, but we were implicated. And the passenger pigeon, we shot every single last one of them out of the sky. So you could argue that where we've played a role in the downfall of a species, and where we have the technology to do something about it, maybe we have a moral obligation. And I think that's an interesting argument. I think it's good, but I don't think it's enough. So when you think about extinctions, people tend to think about things from the distant past. They think about dodos and dinosaurs, but actually, of course, extinction is happening all around us today. We live in a time of mass extinction, where extinction rates are spiking. In the time we've been in this room together, another species has gone. And extinction rates are 1,000 times higher than during pre-human times. So this is our fault. And why it's important is because all living things are part of an ecosystem. And I think of ecosystems as games of giant Jenga. You know, the game where you stack the bricks and you have to take them away? Well, as you take the bricks away, or you take the species away, for a while everything's kind of fine. But then it gets wobbly, and then it crumbles. But because we're in a mass extinction, a period of mass extinction, it's not just like playing a game of giant Jenga. It's like playing a game of giant Jenga with a toddler. It's all going to come crashing down. So it's important. Think about what we might learn along the way. If we could work out how the gastric brooding frog changed its stomach into a womb, we might be able to work out new therapies for stomach ulcers, people recovering from stomach surgery. And if you go for animals that are unique, genetically unique, like the gastric brooding frog, you don't just put a twig back on the tree of life. You replenish an entire branch. So I think, for me, de-extinction is about restoring lost biodiversity. That's the key. It's about putting animals. De-extinction is not about making lonely zoo exhibits. It's about making healthy, genetically diverse populations of animals that could be returned to the wild, where they could interact with their ecosystem in a positive and meaningful way. And that is a tall, tall order. We need to think about what we do very, very carefully. But in my last 5 minutes, I'd just like to mention something else. So if de-extinction is about increasing biodiversity, then maybe it's a form of conservation, maybe. And I've put a cup of chamomile tea here, because when I make this point, sometimes I get shouty tweets from conservationists who get very angry with me. And they say, well, de-extinction and conservation are completely distinct. You can't lump them together, you can't. Because conservation is about helping the living, and de-extinction is about the dead. And I think that is a reasonable point. I don't have a problem with that. But there are 2 points that I make at that. The first is this. If we can get DNA from museum specimens, like this surprised-looking cat, right? Or from mammoth teeth, or from a pickled Tasmanian tiger in a jar, we can get DNA from all these things. If we can take DNA from cells that came from endangered species that have been frozen away, if we can get DNA from that and we can use that DNA to create life, then maybe some types of creatures aren't quite as extinct as others. Maybe there are different degrees of extinctness. And I think that's a really, really interesting concept. So dinosaurs are well and truly extinct. They're never coming back. But we've suddenly got this blurry spectrum between life and death that didn't exist before. And that fascinates me. And the other point is this. Apart from my dog, this is probably my favourite animal on the planet. This is the northern white rhino. And there are just 3 of these animals left. 3. And they live in a wildlife reserve in Kenya. And there's a grandfather, a mother, and a daughter. But they are too old, too ill, and too related to breed naturally. So basically, they are functionally extinct. Here are animals that are alive, but that are the walking dead. They're ghosts. So if we do nothing, they're extinct already. So I would argue that attempts to bring back the northern white rhino count as de-extinction. It is a form of conservation. And there is a guy who works out of Berlin called Thomas Hildebrandt, who is doing some amazing work trying to save the northern white rhino using some of these new high-tech techniques. And I'd like to leave you with one final thought. If we're prepared to think about de-extinction, if we're prepared to at least consider in the future that it might be a positive force, I think, why stop there? How about if we accept, as we must, that the world is changing, that it's warming, that habitats are changing? How about, for example, we use these gene editing techniques that we have to improve the fate of the endangered species on our planet? What about if, when scientists bring back the gastric brooding frog, they add in some genes to make it resistant to the chytrid fungus that would kill it if we released it into the wild today? Maybe that's worth thinking about. Maybe we could engineer fish to be able to survive rising temperatures. And this is another very cute animal. This is the black-footed ferret. These animals currently, all living black-footed ferrets today, are descended from 7 founder members. They're very, very inbred. And they're the subject of a massive captive breeding project. But when they're released into the wild, they die because there's a disease out there that's nobbling them called sylvatic plague. So plans are afoot to edit genetic diversity back into the black-footed ferret. And plans are afoot to edit in genes that confer resistance to the disease that is killing it. And the black-footed ferret could possibly become the very first genetically altered wild animal. It's something that the FDA in America, the American authorities are considering. So there we are, de-extinction. I don't want you to go away with the message that it's about to happen tomorrow. It's not. But the science is moving at a very, very quick pace. And I believe this is possible. And de-extinction won't stop with the birth of the first healthy de-extinct animal. That will be the beginning of a long process that will see these animals nurtured in captivity for a short while. They'll be able to grow up, check to make sure they're healthy, check to see that they can breed naturally, and then, and this is the most difficult part of all probably, released into the wild under the guidance of ecologists, where you would hope they would flourish and interact with the environment in a positive way. So we have at our fingertips these incredible processes, gene editing, stem cell biology, assisted reproduction, that can not only bring back extinct species, they can also sculpt the future of life on our planet. And I'd like to finish with a quote, which I think is particularly relevant at this point. I think it was Spider-Man's uncle who once said, with great power comes great responsibility. So I think we need to consider our next move very, very carefully. Thank you very much for listening.
So we have some time for questions. Brilliant. I'll take the mic. If you could speak into the microphone, that would be great.
Okay. You say that human cloning has understandably been outlawed. Where does the boundary lie? Would we be allowed to de-extinct Neanderthals, Australopithecus, and other such Homo?
It's really interesting, actually. So there's a chapter in the book about them that you just mentioned that includes Neanderthals, and it's very interesting because, of course, human cloning, human reproductive cloning, is widely banned. Not in every country, but in most developed countries, and rightly so. But actually, when those laws were being drawn up, nobody was even thinking about pre-humans. And there are currently, as I understand it, no laws forbidding cloning a Neanderthal because it's not a Homo sapiens. Now, I think, by extension, as we learn more and more about Neanderthals, we're learning that these weren't some primitive savages. These were very sophisticated people for their time. They'd been around much longer than us and were capable of doing some amazing things. They were very, very human, for want of a better word. And I don't think cloning a Neanderthal is a good idea at all. But what's interesting is that we find the technology, in theory, is there to make it happen. And currently there's no legislation in place to stop it from happening. But no scientist in their right mind is considering it, I am pleased to say. And I don't think anybody ever should.
Thanks. Do you think we should have any concerns about a number of hybrid species being produced as a result of gene editing?
Well, I think that's an interesting question. If we start putting genes into endangered species, for a start, we're not necessarily talking about putting genes from another species. We're not necessarily talking about crossing genes between species. It could be that we find some frogs out there that are already immune to chytrid, and we might be able to use that genetic sequence. But in fact, that's kind of a moot point. This idea of using gene editing to kind of enhance wildlife is actually a very logical extension to something that happens in the natural world and in conservation already. So in the natural world, hybridization does occur between species, not often, but it does. Certainly within the bird world, it's really quite common. There was a study recently where they analysed the DNA of a pile of birds and found suspicious genes that came from other species. And then the other thing is that conservationists are very conservative. That's a very good name. And they tend not to like things that are terribly new. But one conservation practice that has been going on for a long time is that when populations of animals dwindle down to very, very low numbers, what they will do is introduce animals from a separate population to introduce genetic diversity. And sometimes that occurs not necessarily within species, but within subspecies. So the Florida panther is kind of a famous example where the Florida panther was seriously depleted in numbers, getting desperately inbred. And then this is a while ago, people imported the Texas panther and they interbred and things got better as a result. So this might seem kind of new and unfamiliar, but I would argue actually this is something that happens naturally and something that one of the scientists said to me, look, a conservation biologist, he said, look, for years and years, people have been solving this problem by moving the animals. I'm suggesting we move the genes. The end result is probably the same. Although maybe if we do it our way, we've got a little bit more control over it.
I have a question. Is anyone looking at bringing back plant life or other fauna, or flora rather, instead of just fauna?
I didn't particularly look at plants in the book, but what we have, of course, are these amazing seed banks, these repositories of seeds where they're trying to harvest as many seeds as they can before plants go extinct. And of course, if you've got the seeds, it's relatively straightforward. So I don't know about de-extincting plant life, but going to the other end of the spectrum where we're thinking about gene editing, of course, GM is commonplace in the arable world now, but the first genetically modified tree is the American chestnut. And that had a gene from wheat pasted into the gene of the chestnut tree because it was suffering from a particular type of blight that was completely hammering this type of tree. And they're now at the stage in the States now where they've got test plots and they're considering about how they might possibly return this tree to the wild.
Do you need DNA from both genders in order to be able to bring a species back? And how many individuals' DNA do you need in order to have enough diversity?
Do you need a male and a female? Yes. So, this depends on us having samples in museums of mixed genders. And if we don't, maybe we think about hybridizing, say, a northern white rhino with its living cousin, the southern white rhino. So you might not make a pure northern white rhino, but you at least save its genes. And then over time, you can breed a purer animal as time goes on. And in terms of a founding population size, we don't know, basically. It depends on all sorts of things, like the generation time of the animal. So the black-footed ferret was presumed extinct. In the 1980s, they thought it had been wiped out by this plague that was killing it. And they went out and caught every single last animal they could find from the plains in Wyoming. They brought 18 animals into captivity. This was 1989, and it was seen as this amazing conservation thing. Nobody had ever gone out there and captured all the members of a species in the wild and put them in captivity. It was really radical for its time. The problem was that from these 18 founder members, only 7 survived. And 9,000 ferrets later, through this captive breeding project that they have ongoing, they're beginning to show signs of inbreeding. But they have cells from some of the original animals that were caught, but that for whatever reason didn't manage to breed. And they figure that if they manage to create new animals from 2 of those original animals, they increase the size of the founding population from 7 to 9, and they think that will give them a decent chance of enough genetic diversity. But it's a question that varies from species to species, and genetic diversity is quite a stochastic, random process. So it has a lot to do with the random shuffling of the genes when creatures reproduce. You might be lucky and get away with a very small founding number, or in some cases it might take a larger one.
Okay, nice talk. Thank you very much.
Thank you.
I have a question. So you talked about there's a lot of technology to recreate animals that are extinct. Do we have that amount of technology to predict the impact of this animal in the environment? Because if we bring back the mammoth and we put him somewhere that would be nice for him, what can we expect from him destroying everything else because there's an imbalance there? Or like the pigeon, because we have pigeon problem, right? There's lots of pigeon. If we put a new pigeon there, what can happen? Do we have technology to think about it, to understand what might happen with these dead animals coming back? And just one other thing, do you think that if someone finds a very big piece of dinosaur like they found from the mammoth, maybe they can come back? That would be awesome. Like Jurassic
Wishful
Park.
thinking. All right, well, so the first question. What de-extinction at the moment is a process in development. It's kind of at the cell biology stage and at the create an animal stage. And what's missing, in my opinion, is this kind of long-term thinking where we start thinking about what happens when these animals are released. And that's not to say that the thinking's not going on, but that's a discussion that's really just starting now. And we need ecologists brought on in these discussions from a really early point. And not only that, we need paleoecologists. So we need people that have studied these animals the first time around. They've looked at their remains. They've learnt from what happened when this animal was around the first time around. And certainly with the passenger pigeon, that's something that's happening. The guy who's leading the passenger pigeon project is a guy called Ben Novak, and he's passionate about the project. And he's gone through all the historical records. And he has a very, very clear idea of how passenger pigeons, when they were alive, interacted with their environment. So they were like this swarm of feathered locusts. These birds would land and devastate the deciduous woodland where they landed, which doesn't sound great, right? But bear with me. So they would land en masse, they would topple trees, they would cover the forest floor in guano. But from that apparent devastation came life. So a stagnant, closed canopy woodland turned into one that was sunlit. And the guano fertilized the ground. And it paved the way for the growth of all sorts of different plants, which brought in insects and small mammals and amphibians, which brought in big grazers, which brought in predators. And he's modelled this whole thing very, very carefully. And he is pretty confident that because the passenger pigeon was such a keystone species, its actions had these repercussions across the entire landscape, that if you introduced it back into that environment, it would drive this recycling of the eastern North American forest that has been absent since it's been gone. But that's something we need to think about really carefully. You know, ultimately, we don't know. And ultimately, we need to build that into the release of these animals. We also need to think about a get-out plan for these animals. You know, the world is riddled with invasive species that are causing, they're responsible for most of the extinctions that are happening, certainly on islands. They're a big, big problem. You don't want anything that you go to all this trouble of creating to be considered a pest. You don't want to have to kill it again. So these are things that we need to think about really carefully. And I hope that answers the first question. And the second question is, with dinosaurs, there's a lady called Mary Schweitzer in America who is, she studies dinosaur bones, and she's seen these really tantalizing glimpses of biological molecules inside dinosaur bones. So I wouldn't call them proteins, but she sees signs of proteins. She sees signs of cells. And her results are pretty controversial, and yet they're beginning to be backed up by some other groups. There was a group at UCL last year who found something similar. People haven't really looked in dinosaur bones for these kind of biological molecules because we've been taught at school that they are inert mineral, that they're inorganic. But that dogma is potentially beginning to change now. And she went even further, because for her, of course, the natural progression is to say, could DNA be in there? And she ran some tests on some quite ancient dinosaur bones and sees signs of DNA. So I'm not saying there's DNA there, but she saw a signature of DNA. However, it's one thing for DNA maybe to be there in fossils, which is questionable, fossils of that age. It's quite another for it to be in any usable form. And I don't think we will ever get DNA in a usable form from dinosaur bones. You have to be able to reconstruct the genetic sequence, the genome, to give you that set of instructions to create life. And would you like a pet dinosaur? Me too, but sadly, we're going to have to settle for dogs and bring them in. It'd be better bringing in dogs to work than dinosaurs. That could get messy.
One here.
Hi, thanks a lot for your talk.
Thank
Really
you.
interesting and engaging. I have one question. It seems like the biggest problem is having a source of DNA
Yep.
and from endangered species. Are we doing anything to create a sort of bank for the future? Because if it's not today, it might be in 100 years. These techniques will be finalized and work. So are we thinking about building this instead of relying on single museums that might have one species DNA or something like that? Are we doing something more global?
Yeah, so we've got this massive repository of DNA in museums. But it's much better to have frozen cells, because then you've got something that is alive that you can start with. It's far easier to start with DNA inside a living cell than DNA inside a stuffed museum specimen. So the museum specimens are kind of like a source of last recourse, if you like. What is really important are the viable cells that are frozen in vats of liquid nitrogen. And there are multiple initiatives all around the world that are doing exactly what you're thinking of. The biggest one is at the San Diego Zoo in America, where they have something called the Frozen Zoo. And through their zoo there, but also through many other zoos and wildlife collections around the world, they send body parts to San Diego Zoo, and they culture these cells and they freeze them. And I heard the guy who leads this is a guy called Oliver Ryder, and I heard him speak last year, and he says he's always getting interesting things in the post. So apparently there was some orca at SeaWorld or somewhere that had just given birth, and they grabbed the umbilical cord and they packed it in dry ice and they sent it to him, and they cultured cells from that and they froze them down. So yes, and that's the interesting thing, that if we have these frozen cells in liquid nitrogen, you have a species maybe in stasis, not quite alive, not quite dead. I mean, clearly it is dead, but it's not as dead as some other things. So yes, people are trying to collect as much biodiversity as they possibly can and squirrel it away for a rainy day. And it's not just there, actually. I should say the main point of these things is not really there for bringing animals back to life. The main reason for these cells being collected is to use them as a source of learning. They're used in research papers all around the world. But by studying DNA from endangered species, you get an idea of the health of these endangered species. You can see if they're going through a genetic bottleneck. It can give you some idea of how to manage your captive breeding programs. So the cells that are being frozen away are being used for all sorts of excellent purposes. And de-extinction is maybe just a glint in the eye somewhere off to the side of that, but it could be used for that in the future.
On the same lines, what's the difference then between editing an elephant to be like a mammoth? Surely that's the same, along the same lines. Is it just going to be a woolly elephant?
It's going to be a mammothant or an elemoth. No, you're quite right. It won't be. If George Church has his way, he's not calling it a mammoth. He's saying he's making a cold-adapted elephant. He's making an elephant. His creature, I should also say, none of these creatures that get de-extincted, if and when it happens, will be the same as the originals for many, many reasons. Many reasons. One of the reasons is their genetic makeup. So George Church's is an extreme example where he will end up, if he succeeds in his quest, with an animal that is genetically overwhelmingly elephant, with a little sprinkling of mammoth DNA. But he says, well, hey, I'm completely fine with that, so long as this animal acts like a woolly mammoth and can perform the same ecological role as a woolly mammoth when we put it back, say, into Siberia, then I don't have a problem with that. And that's the way the de-extinction community feels. It's not about creating an absolute replica. It's about something that will fulfill a lost ecological niche.
I had a really quick question about the pregnancy, or the surrogacy, rather. Say, for example, with the Picardo goat, how do you select the animal to kind of carry the Picardo goat to term? Is it a similar mountain goat with, that shares a lot of the DNA, or also for the woolly mammoth, would that potentially be an elephant, or how does that process?
Yeah, what you need to do is you need to find the closest living relative. So, I said it's great if you can bring back an animal that is genetically distinct. Well, of course, the problem there is that if it is genetically distinct and unique, what do you use as a surrogate? Which is where something like a frog is great, because you don't need a surrogate. You could, if you don't need a womb, you've removed a whole lot of problems. Now, with the Picardo, they used, obviously, an existing type of goat. They had to create a particular hybrid, because the Picardo's closest living relative ended up being really flighty and difficult to look after in captivity, and it literally climbed the walls and was so neurotic they couldn't work with it. So they created a hybrid goat where they bred its closest living relative with a domestic goat and found that worked really well as a surrogate. So I mean, when I give these talks, you don't get a flavour of the huge amount of research and effort that goes into what happens. So just choosing the right surrogate and getting it to work was massive for the Picardo team. And yet, with the woolly mammoth, my big problem with the woolly mammoth is that it involves using elephant eggs, which are in really, really short supply, and elephants as surrogates. Now, there is one potential way around getting hold of elephant eggs. Scientists are now in this position where they can take skin cells, reprogram them into stem cells, and use them to generate egg and sperm. Right, so that potentially is a very exciting tool we have at our fingertips. It's been demonstrated in mice. The whole process hasn't been shown particularly for endangered species, but people are working on that. So maybe we can get hold of a load of elephant eggs to perform these techniques on. But then you still need a surrogate, and I don't feel good about that. Elephants are seriously endangered. They're not experimental animals. And George Church, who is amazing, will say to you, come on, think bigger, think artificial womb. Until we get to that point, or until the elephant's out. Do you know what? I'm never going to be comfortable with using an elephant as a surrogate. For me, that is a massive stumbling block with a woolly mammoth. And we need to think about the repercussions of we can't sacrifice one endangered species to bring back another from the dead. So all of these questions need to be thought about really deeply.
With that, please join me in a massive round of applause for Helen Pilcher.